Hello, good morning. I am going to discuss the rest part of the cell, centrosomes and centrioles. Centrosomes is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of nine evenly spaced peripheral fibrils of tubulin proteins. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked to each other. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplet by radial spokes made of protein. The centriole form the basal body of cilia or flagella and spindle fibers that give rise to a spindle apparatus during cell division in animal cell. Now, nucleus. Nucleus as a cell organelle was first described by Robert Brown in 1831. Robert Brown discovered the nucleus in 1831. It is dye under observations of microscopes with the help of the dye chromatin by Flemings. The interphase nucleus, that is nucleus of a cell when it is not dividing. So this phase is known as interphase nucleus, has highly extended and elaborate nucleoproteins. Fibers called chromatins. This is known as chromatins. This is known as chromatin. So here showing euchromatins and heterochromatins, both are known as chromatins. Nuclear matrix, nucleoplasm, and one or more spherical bodies called nucleoli. Electron microscopic has revealed that the nuclear envelope, this is nuclear envelope you see in the diagram, outer membrane, inner membrane or perinuclear space, that is constitute to form the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nm, called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm, and this structure is known as endoplasmic reticulum. Endoplasmic reticulum. Endoplasmic reticulum. The outer membrane usually remain continuous with the endoplasmic reticulum. and also bear ribosomes, and these structures is also known as ribosome. These are ribosome. That means on it. At a number of places, the nuclear envelope is interrupted by minute pores. These are the minute pores, like this. Interrupted by minute pores, and which are formed by the fusion of its two membranes. These nuclear pores are passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. This is the way by which RNA and other proteins exit from nucleus to cytoplasm and enters cytoplasm to nucleus. There is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Erythrocytes of many mammals and sieve tube cells of vascular plants have minute cells which lack the nucleus. The nuclear matrix or the nucleoplasm, these are the nucleoplasm. Here the name nucleoplasm. The contents of nucleolus is continuous with the rest of the nucleoplasm. As it is not a membrane bounded structure, it is a site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in the cells actively carry out protein synthesis. The interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of the cell division, cells show a structure called chromosomes in the place of the nucleus. So these are the structures of chromosomes. Structures of chromosome with kinetochore. Kinetochore. These are the nucleus. These are the chromosomes. In chromatin contains DNA and some basic proteins called histones. Some non-histone proteins and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 chromosomes. Every chromosome essentially has a primary constrictions or the centromeres. Primary constriction or chromosomes. You see the diagram. These are the chromosomes. Primary constriction or the centromere. These are centromere. Centromere. On the sides of which disc-shaped structures called kinetochore, and this disc-shaped structures is known as kinetochore. These are the kinetochore. Based on the positions of the centromere, the chromosomes can be classified into four types. First is metacentric. Four types of chromosomes. Metacentric, metacentric, in which the middle centromere forming two equal arms of chromosomes. So these are the structures of metacentric chromosomes in which two arms are equal to each other. Second is submetacentric. Submetacentric, in which submetacentric, metacentric, in which chromosomes has centromere slightly away from the middle. You see, slightly away from the middle, and in which both arms are equal. So these are the metacentric, and this is known as the submetacentric. That resulting into one short arm and one long arm. Third is acrocentric. Acrocentric, in which one arm is short, chromosomes, acrocentric chromosomes, in which The centromere is situated close to its end, forming one extremely short and one very long arm. So these are the position of acrocentric, and that is the fourth is telocentric. Telocentric, in which the centromere is on the positions and the two arms is very small and one is very large. That is A, B, C and D. Metacentric, submetacentric, acrocentric and telocentric has a terminal centromere. These are the terminal centromere. So these are the four different types of chromosomes on the basis and positions of the centromere during the process of cell division. So these are the structures and functions of nucleus. Sometimes a few chromosomes have non-stem secondary constrictions at a constant locations. This gives the appearance of a small fragments called the satellite. So this structures is known as the satellite. Satellite. Satellite, and it is also known as the secondary constrictions. Secondary constriction. Short arms. These are the short arms. Short arms. And this is the large arms. Large arm. And this is centromeres. So types of chromosomes on the basis of and position of centromeres. Types of chromosome. Types of chromosome. Microbodies. Many membrane-bound minute vesicles called microbodies that contains various enzymes are present in both plants and animals. So, your complete cell, the unit of life, its organelles and its functions are so in various videos. On the basis of this, the cell is also known as a structural and functional unit of life. Thank you.